And I guess it's still morning, huh? Good morning, everyone. So this is our aging seminar series for resilience and independency. We're so glad to have our scholars to present their work today. So my name is Zhao Chen. I'm professor and department head in the College of Public Health for EP and Biostat. So I'm going to be hosting this seminar and monitoring the presentation. Before we get started, I'd like to welcome Dr. Janko Niklas to give a few words about our training program.
Thank you, Zhao. My name is Janko Nikolic. I'm department chair of Immunobiology and co-director of the Center on Aging. Mindy Fain is the other co-director, and she is in clinic and couldn't join us today. Might drop by later if she has a chance. With all of you being in the program, we're really happy that we have assembled a super talented group of young investigators who are doing great things. We were fortunate to get institutional support to start this program. We have, as you know, gone through a PEPPER grant application that we feel was a great first step, but this is one of those that we know that we have to take several steps in order to eventually be successful. We got our reviews back from that, and we're trying to see what is the most productive way forward from there. But this research, translational research on aging, basic research on aging, remains a focus of the university at many levels. It is a priority for the Bio5 Institute. It is a priority for the strategic plan and several sub-initiatives within the strategic plan. And it is a priority for the College of Medicine as well. So we'd love to hear what you guys have been able to do during the pandemics. We understand that it was a tough time for everyone. But what we do now and how we set it up is going to go a long way towards how successful we can be. And we'd love for you guys to be successful. So I don't want to belabor anymore. We have a whole bunch of great talks for today. So let's turn it over to the presenters.
Thank you. So first presenter will be Dr. Wing Tang.
Good morning, everyone. And hi, everyone. My name is Wei Tan, and I am a vascular surgeon with interest in health disparity research. And I'm really grateful for the support for the program, which I use to look at frailty and racial and ethnic disparity for patients undergoing surgery. There's some challenges, especially the pandemic, but I think we make pretty good progress with the projects. So our population is aging, there's no question. There are more and more people that are undergoing elective, semi-elective, and emergent surgery. And this is an editorial from New York Times, which sum up the challenges that we have. The elderly are getting more complex surgery, and they don't end well. We now know that frailty is an essential assessment that we have to make as a surgeon, as well as subspecialties to look at our patient before surgery, because we know now people who are frail, elderly or not, are more likely to have complication, death, increased hospital length of stay, and reduced quality of life. These are a paper from Dr. Balal's group as well as my group that demonstrated that frailty is an important assessment for people undergoing surgery. So for example, for people undergoing carotid surgery, people who are frail, independent of their medical comorbidity, were more likely to have stroke, MI, and death. My interest is in health disparity. So there's no question that racial and ethnic minority experience worse outcome for a variety of surgical procedures, including cardiothoracic surgery such as CABG, open aneurysm repair, and other vascular procedures. As a faculty for SALSA, which is Southern Arizona Limb Salvage Alliances, my practice focuses on limb salvage. That means to prevent amputation. So people, minority, racial and ethnic minority, when they undergo procedure, they have worse outcome and they experience more amputation. Why is that relevant? Because minority are growing across the country and especially here. In fact, Hispanic is the largest minority group in the country right now. What we don't know is whether there is any impact between frailty and disparity in surgical outcomes. We know that from patient level factor, for example, insurance status, for example, medical comorbidity, societal level, for example, where they live, or policy level, for example, whether they live in Medicaid expansion state, are associated with disparity in outcomes. But we are not sure yet whether there's impact of frailty, whether we can explain that based on frailty. So for this project, our hypothesis is when we compare to white, Hispanic, and other minority, age 65 and older are more likely to be frail, and they have highest risk of complication independent of their frailty status. So there are two aims. Aim one is we try to describe and measure the impact of frailty phenotype and disparity. And second aim is to try to identify people where we can allow for early intervention to try to reduce the disparity of surgical outcomes. So for aim one, we used the American College of Surgeons NIST database from 2014 to 18. We included patients aged more than 65 who underwent procedure to improve blood flow, so the revascularization procedure for the leg to prevent amputation. We classify whether they are frail based on the verified modified frailty index. And we look at outcomes including death, mobility, and readmission. We have 9,000 patients. The mean age were 75 years old, 63% were male, 70% white, 20% African-American, and 7% Hispanic. Based on this study cohort, approximately 24% of patients were classified as frail. However, African-American and Hispanic were significantly more likely to be frail when we compare to white, 31% versus 70% with a p-value of 0.001. When we do multivariable analysis, after controlling for age, gender, American Society of Anesthesia classification, comorbidity, and indication of procedure, we know that compared to white, African-American were 1.9 times more likely to be frail, Hispanic 1.2 times more likely to be frail, independent of their age. For frail patient, when we compare to white with frail status, African-American and Hispanic were significantly more likely to have complication, including 30-day complication and readmission. Interestingly, when we look at non-frail patients, the disparity among the outcome were actually not too significant. So majority of racial and ethnic disparity in surgical outcome following vascular procedure are in frail patients. This is a publication that just came out recently. We look at people that fail procedure and need amputation. Similarly, minority were more likely to be frail and they're more likely to have complications. For aim 2, we plan to use the UA Age-Friendly Healthcare System Initiative with support from Dr. Fain. We just completed data collection and in the process of data analysis. And the aim for this is to try to identify risk factors for people who have adverse risk surgical outcome, as well as to try to identify group where we can target for early intervention. So in summary, race and ethnicity actually influence frailty status in vascular patients, especially among those undergoing peripheral vascular procedure. And we aim to try to identify potentially modifiable risk factor and biological markers to address the impact of frailty and disparity in surgical outcomes. And thank you for your time.
Thank you very much for the presentation. Now we can take a couple of quick questions.
Yeah, do you prefer people to do it through the chat, or we're a relatively smallish group? Shall people just ask directly, or?
Yeah, you can just unmute yourself, ask a question directly. That will save time.
So Wei, can you describe a little bit the modified index that you're using to measure frailty? It's an 11-point, you said, right?
Yeah, so it is verified and used by our group, including and Dr. Balas's group too. I think we are one of the few that initially use it, and it's verified across the group, and it's verified for the NISCRIP database. So we use different factors, including age, gender, comorbidity, and as well as, so these are all retrospective actually database based on a variable and not prospectively collected. So that's why we propose to do AIM2, where we use the prospectively collected data to try to understand the impact of frailty and the disparities.
Right. Yeah. That I know that Bilal's group had used a modified 15-point sort of like surgery-related frailty index, which is, of course, both of these are really modifications on the Rockwood as the basis. So yeah, that's. I think we'll talk to you maybe. If you could email me and Makiko what are the parameters, it would be interesting from our standpoint, because we usually do freed on most of our patients, and then when we correlate it with with what you guys have, it would be good to have parallels. We could modify some of our procedures also to use it. So when you do prospective, what would you do then?
So there are different way of doing it, but for AIM2, it is done. So what happened with the data that we're using is the patient was sent to see Dr. Fain, and I think they assign whether they are frail based on the same variable that we use in addition to the American Geriatric Society parameters. So it is assigned prospectively, which is more accurate. There are a lot more publications out there that suggest that just by eyeballing patient and assigning a status is pretty accurate. I don't know whether you have seen those publications. Usually if you can tell they are not frail, they are usually not frail. And if you say they are frail, they are frail. I think the argument is in between, where those are intermediate people.
Thank you.
Okay, thank you for the question. We're going to move on to our next presenter, and Dr. Ann Scholas-Ray.
Hey, good morning. I'm going to share screen. Okay. So my project focused on pro-resolving lipid mediators and how they play a role in resolution and how that relates to resilience and frailty. And I think this group can appreciate that inflammation drives a lot of different diseases. And we typically in my research group look at heart disease, but we're very interested in the role of unresolved inflammation in driving disease. So a lot of our research focuses on using dietary supplements like fish oil that contain EPA, DHA, and DPA in order to increase tissue levels of these omega-3 fatty acids so that when we have an inflammatory challenge, we can produce pro-resolving lipid mediators termed resolvins, protectins, and mucins. And specifically, these are all using EPA and DHA as substrate, these compounds that we're very interested in. And there are intermediates in that pathway. This is going to be important a little bit later in the presentation, but 18-HEAP derived from EPA, we've been calling a precursor or a pathway marker for the production of resolvins. And for DHA, we can look at levels of 17-HDHA, which is a product of this compound, and then 14-HDHA. But these compounds are very important because in the absence of having resolvins, protectants, lipoxins, and other pro-resolving compounds, the inflammatory response can get stuck in this initiation phase, and there's no switch to resolution. So for the pilot study, we hypothesized that the capacity to produce these lipid mediators would predict both frailty status at baseline pre-surgery and then post-surgical complications. And this would be independent of biological age. And this would give us a way to sort of compare and contrast the responses of older adults with younger adults. And the way we proposed to look at that was to first start with our data set and our samples from younger healthy adults to characterize what is a healthy pro-resolving response. And so to do this, we elaborated analyses of 20 healthy adults who were injected with endotoxin intravenously. And we have a time course response. And then we wanted to see whether there were specifically lipid mediators or a cluster of lipid mediators that would be predictive of post-surgical complications, as well as baseline health status. So I mentioned we have this model of induced inflammation. This is what it actually looks like. This is an IV that has been placed. There's another IV for saline administration, blood sampling, and we had access to these single-use vials that are regulated by the FDA through the IND process, and we inject a weight-adjusted dose of LPS directly into the vein, and then follow the time course of response out to 5 to 7 days after injection, depending on the study. And what we see with that model is a very consistent and robust inflammatory response. So on the days that the participants were injected with LPS in this crossover study, we got this nice CRP response that starts coming up at 6 hours post-injection and peaks at 24 hours. Whereas when participants were injected with saline, and this was blinded and crossover, so these are the same people, but with the injection of saline, you see no such response, nice flat, healthy, no inflammatory response. So we know that this model works even on an individual basis. Every single participant had a response. And again, these are young, healthy men for this model and this inflammatory response. This also is true of the cytokine response, that we get a consistent and robust response. So here you have IL-6 and TNF-alpha response with the LPS versus saline. You can see that's earlier. And that corresponds with the symptoms that participants experience too. Okay, so we use this model then now that we're happy that it's a very consistent and robust inflammatory response that we can look at. We use that to look at whether omega-3 fatty acids would change that response. So we gave people the prescription dose of omega-3s, which is a high dose, more than 30 times what the average American would consume on a daily basis. And we did this for at least 8 weeks in a crossover design, again, randomized, blinded. And then we did an endotoxin challenge at the end of each treatment period. And what we saw was absolutely no effect on the protein response to this inflammatory stimulus. So the CRP response for the fish oil versus the placebo right on top of each other, which says to me that we did not suppress the necessary and protective acute phase response. And I think that speaks to the safety profile of omega-3s, but we did not knock down the IL-6, TNF-alpha, or CRP response in these healthy adults. So we've preserved the acute response. However, I mentioned we were really interested in these compounds that drive that switch from pro-inflammation to pro-resolution. And what we did see was a robust and very easily measured increase in 18-HEAP, which is that pathway marker for production of resolution products. So it increased in response to omega-3s, it increased further in response to the inflammation challenge, and then it comes down at 24 hours. And we also see different responses for the DHA products. This was 17-HDHA, doesn't do the same thing as the EPA product, and neither does the 14-HDHA. So again, we're interested, and the 18 heap really, came out as the winner, is sort of the way the time course looked was very nice for us. But I want to say, I mentioned these are thought of as sort of pathway markers or precursors, but they have their own effects to regulate inflammatory responses. And this is a growing field, and my PhD student is currently really digging in and summarizing this for a review article that we're working on. So what we were hoping, or we were surprised to see in a preliminary analysis that we were expanding on, is that plasma and serum have very different lipid signatures. So the only difference between plasma and serum collection is that serum's allowed to clot. But in the plasma of people who were inflamed, provoked inflammation, we got this nice increase in the blood, in the plasma, of this 18 heap that we're really interested in. However, in serum, we can see high production at baseline when blood is allowed to clot, and then suppression during the acute inflammatory response. And I think that's really interesting because by allowing blood to clot, that's sort of a ex vivo inflammatory stimulus and could reflect sort of the maximum capacity to make these compounds. And so we were thinking this could be predictive of post-surgical outcomes related to resolution of inflammation. So what we established with this is that our omega-3s aren't changing the inflammatory response, and we can increase these precursors in the blood by supplementing with omega-3s. And when we went to look at serum markers a little bit more, a different lab got different results. So now we're working out why this serum response in this lab was different than the ones that we got. The other interesting feature of these compounds is that they are carried on lipoproteins. So whether you're measuring the free or the esterified pool actually changes interpretation as well. But we still feel strongly there's a way to use these as a sort of resilience index. Both the provoked response in result of a surgical challenge or illness vaccine, as well as allowing blood to clot or doing some other sort of ex vivo manipulation to see how much of these compounds is produced. So again, this was sort of the first part, but then we are moving into using that line of reasoning to predict post-surgical complications now that we've established what a good response looks like. And so to do that, we were looking at a time course of a banner schedule of patients who were undergoing emergency general surgery for acute appendicitis and cholecystitis. But I will say we had a ton of technical difficulty getting into Banner, and just some of the challenges I think of I'm on main campus in nutritional sciences. And so my team had a lot of administrative barriers, and a lot of people tried to help us, but in the end, it became very difficult to coordinate getting access where we needed to, and all the moving parts of Cerner and knowing when the surgeries are scheduled, getting to people, getting the follow-ups. So we didn't get the 10 people that we were hoping to with our pilot study. So I think that's moving forward. We'll have to, I think, figure out something that from a logistical standpoint works better for this question. But, and I think at the start we talked about giving updates on kind of all of our status, and I'm one of the clinical researchers who was just shut down completely because I do intervention studies, and they are essentially bringing people into campus, which is high risk. So all of our studies were terminated, and we're setting up a new research facility off campus to minimize risk to participants. Okay, I think I'm at time, so I'll take questions.
Perfect. Very interesting finding. Very promising moving forward. Any questions? I know probably Yanko is ready to jump on. This is your area of expertise.
Yeah, it is provocative. You know, so I was talking to Ski about some of this, and you know, he was saying that in general, heparinized plasma is less of an issue analytically anymore, but obviously not for your compounds, right?
Yeah, so one of the things, when we're digging in and trying to get a good clinical summary of the literature is that we found that people will use plasma and serum as though they are the exact same thing in the papers. They'll even go back and forth. Yeah, and we know, and this is well established, not just us, multiple groups have shown that if you allow the blood to clot, the cell-cell interactions and activation of the immune cells and the platelets, it completely changes the profile of these compounds. And if we can harness that to get information, that's useful, but that is a barrier that, not even drilling down to the specific of heparinized versus EDTA plasma, people aren't even delineating between plasma and serum in their analysis. And the thought is that with serum, everyone has said that, the levels are higher, but the tone of it is such that it seems like there's going to be like a multiplier. Like, so to convert plasma to serum, you just multiply everything by 10. But I hope I showed with that time course that it's not a constant adjustment. In the context of acute induced inflammation, the serum levels are plummeting and the plasma levels are going up. So the literature is sort of flooded with a lot of cross-sectional analyses that aren't really controlling for health status and drawing inference and off of blood concentrations and X outcome. So
your 18 HIPI levels, really, the dichotomy between serum and plasma, was that also, did that affect any of the other inflammatory markers?
So I think the 17-HDHA might have looked a little bit different, but if we were to do that for all the protein markers, we'd never see anything like that. And I didn't go into it because of time and just there's a lot of already details, but we couldn't detect the actual resolution compounds that most research focuses on. So the things that the 18 HEAP is a precursor for aren't really detectable in blood the way that marker is. And that's been sort of a dicey thing to navigate because it's very detectable in sort of enclosed like pus or tears or breast milk, but in general circulation, it's just not, and not everybody likes that interpretation. It's like one of those controversial things that people get really heated about for whatever reason.
Yeah, well, inflammation is supposed to be local, hopefully, although, when you have sepsis, it isn't, or, a couple of other conditions. So measuring it locally would be really cool. I just don't know how you would do that, So your LPS injection is IV? Yeah.
And I thought that was really interesting too, that CRP didn't come up until 6 hours. That was another, so healthy people. And I had it done to me before I recruit anybody, just because, again, I don't like to, I want to know what it feels like before I try to tell other people that it's safe. And it really is that 4 to 6 hour, first 4 hours, 2 to 4 hours specifically, when all the side effects, when the TNF alpha is up. But it is systemic inflammation. You get the same feeling as like getting a viral infection, any sort of infection. I think it's very analogous to that. The first few hours when you're coming down with the flu or COVID or whatever, you get that just sort of like, oh no, and then it just lifts. So we did get tons of 18 HEAP in the systemic circulation, and so that's encouraging. And that's actually, no one's really, the focus has not been on 18 HEAP. It is reported in papers, but more as sort of a tag along to the rest of the story. So that's why we're trying to get all that information together.
Yeah, very intriguing discussion. So I hope you will do that offline, continue the discussion. I just wanted to know more. Okay, so consider the time, let's bring up our next presenter, Marv Sleppy. You're the next, Dr. Marv Sleppy. Do we have him?
I don't see him on.
I know he's giving the founders talk today at the College of Medicine, so
maybe he might have his hand full.
Okay, so if anybody have a way to reach to him, so can we go to the next presenter? Just keep it rolling. Let's see who is the next. Is Georgia, Dr. Georgia Suchby?
Yep, I'd be happy to go next. Slides up here, just a second. All right. So thank you for having me. I'm an assistant professor in molecular and cellular biology. We're over in the BSRO building in Bio 5. And the project that we're looking at for the center is looking at this metabolite 3-hydroxyanthranilic acid, or 3-HAA, which is a tryptophan metabolite in the Kynurenine pathway. And we're looking at using elevation of physiological levels of 3-HAA to improve sepsis recovery in aged mice, which is the main topic of this. This project has been delayed somewhat by the shutdown, the research shutdown, so we're not quite as far as we like. But I'll show the preliminary data that we have on our mice. And then in parallel, we have some work going on in C. elegans looking at the same pathway and immune response. And so I'll talk about the kind of parallel work that's going on in worms, so you can do things a little bit faster in worms. So there's that data is a little more advanced, and we're learning something about the mechanism that I think is relevant to the mouse work as well. Let me see if I can get this to advance. There we go. Just a brief slide of background. The way we got to this pathway. So the pathway is the main metabolic pathway for tryptophan. About 90% of tryptophan goes through this pathway called the Kynurenine pathway, which terminates in the production of NAD. And about 90% of the tryptophan we ingest goes through this pathway. It's immune responsive, particularly this first step, IDO or TDO, gets upregulated in response to inflammatory signaling and immune signaling. And so this pathway gets heavily turned on during inflammation. And all these other processes that perhaps are somewhat more familiar, generally translations, serotonin production, tryptamine metabolism, these take up maybe 10%. You see estimates vary from like 2 to 10% of the ingested tryptophan go to these other processes, but really the majority is going through this other one. So we came across this pathway through a screen and identified this gene, KYNU, where we knock it down in the worms and we get a big lifespan extension, about 20%. And we've actually done a lot more work and identified 3 points in the pathway. This first point, TDO2, KYNU knockdown or HAAO knockdown, that'll give us a kind of robust lifespan extension, the biggest of which is knocking down HAAO. And what we think is going on with HAAO is that when you knock down this enzyme, the 3-HAA builds up, and we can replicate the HAAO lifespan extension by just feeding the worms 3-HAA. And if we start to look through the literature, we've really been focused on this HAAO and 3-HAA interaction. 3-HAA has some evidence that if you elevate it in autoimmune models in mice, or in other inflammatory models like asthma, you can get an improvement by elevating 3-HAA, either by inhibiting HAAO or increasing or just directly supplementing with 3-HAA. So that it looks like there's some sort of benefit to kind of inflammatory disease. And so since we're interested in aging, the kind of goal of this project was to look at an age-associated immune challenge, sepsis in this case, so kind of the hyper-inflammatory response you get to a severe infection. And if we can kind of tamp down that inflammation. So sepsis is one of is a major cause of mortality in elderly individuals who are admitted to hospitals. And it's one of the more expensive things that are dealt with by hospitals. And so it's kind of a need to deal with this problem and find ways to help improve survival and health following sepsis. And so we wanted to look in mice. We're using young 6-month-old and 20-month mice. This is just an overview of what we want to do. We're using 2 models of sepsis. The work I'm going to talk about today is just injecting bacterial LPS from P. aeruginosa. And this gives you kind of the inflammatory response without the pathogen being present. So this part of the work is specifically looking at that. And then we follow up with treatment with vehicle, either direct 3-HAA or inhibition of HAAO with this tryptophan analog, 6-chlorotryptophan. And then we're looking at survival in one group. And then we haven't gotten here yet, but we'd like to look at cross-sectional studies. And I want to note the doses here are kind of pulled out of the literature for the LPS and for 3-HAA and 6-chlorotryptophan. And some of the early work we're finding is maybe the doses that are published are not exactly what we want to look for. And so this is just kind of the preliminary mouse data from what we're looking at initially. And this is where I think maybe our dosing is a little off. So we've done a few studies. We started with a small group of 24-month-old, so old C57 black 6 mice. We hit them with 2.5 milligrams per kilogram of LPS, and then followed up with an injection of one of these drugs, 4 chloro-3-HAAs, another HAAO inhibitor, at 200 mg per kg, which is where most of the mouse studies are hitting for these guys. And we got kind of that, we did not see an improvement in survival. The mice lived maybe 3 to 4 days on average in the wild type, and we actually maybe had a slight shortening in lifespan. And so we're actually suspecting now, for a couple of reasons that I'll go into, that maybe our dose for these drugs was a little high. This is a very high dose for an additive, and maybe it's too high for these kind of severe septic situations. And we have followed up in young mice to start to look at dosing, and we can see if we pull this back from the 200 mg per kg, we do a little bit better in young mice with a little bit lower dose. And I think we probably need to go a little bit lower. One of the problems with this study is, so I mentioned the 3-HAA dosing, but the LPS dosing is, we're also think we need to kind of hone in on a little bit better for this model. One of the things we see is that one of the major driving factors is the biggest survival difference in the mice in these groups was actually driven by sex and or body weight, which are confounded in this case. And so we've been giving them a dose per kilogram. And what we think we maybe need to do is just stick with a single, it may be the total amount of LPS given, not the dose per weight that may be important. So we're actually kind of pulling back and looking at a dose response to really get this model working the way we want it to. The kind of hope that we have for this situation is we've been breeding both KYNU and HAAO knockout mice for kind of a general aging study, and we're only looking at the homozygous knockouts there. So we have extra heterozygous mice coming out of that study, and we decided to look at what happens if we give an LPS dose in these heterozygous mice. And we have both the KYNU, which is upstream, the direct upstream enzyme that produces 3-HAA, and HAAO, which is the enzyme that typically breaks down 3-HAA. And what we see in this preliminary study of maybe 5 to 8 mice, I think is what's in these groups, is the HAAO mice do very well, even though they're only heterozygous. So these should have some elevated 3-HAA, but not dramatically elevated, whereas the KYNU don't do as well. And so we have some hope that, like, if we can get the 3-HAA levels down a bit, maybe that we're just not in the optimal dosing range. And so we want to continue playing with these knockout models, and also try to get our injection dosing down so it's maybe more replicating what's going on in these mice. And this is still very preliminary, it's small numbers, so we need to replicate this with some more mice, but this is looking hopeful in any case. The other side of the dosing equation, where we think we're not quite in the optimal range for sepsis, is through the Jackson Laboratory, we're running a different pilot study where we're looking at lifespan in old mice. So these are mice aged out to about 2.5 years. And at that point, we initiate, so this is not an injection, we're giving them diet supplemented with 3-HAA. And this is the dose per day is around 50 to 500 milligrams. So this is still high, but it's a dietary dose. So the effective dose getting into the mice is going to be quite a bit lower than in the injections. And this is still, again, very preliminary, but we start them late in life, and then we're seeing a separation of these lifespan curves. And it looks like the low dose 3-HAA diet may be the one that's giving us the best. So we need to have these curves go out a little bit more. But again, there's hope for kind of these lower doses of 3-HAA being kind of the optimal range. So that's where we are with the mouse study. We're again taking a step back to really nail down our doses, and then we'll go forward with the full sepsis study again. And we're fully back up and running now, so hopefully that will be very quick to come out. I want to shift to C. elegans, where we have maybe some more information. So what we're doing in this model, we're feeding the worms Pseudomonas aeruginosa. They normally eat E. coli, and Pseudomonas is kind of shows this age-dependent toxicity. So worms normally live about 3 weeks, and if you on E. coli, but if you put them on P. aeruginosa, they're down to like maybe a little over a week for the median lifespan. And the survival has an age-dependent effect. So if you expose the worms at different ages, you get a shorter lifespan. And what we find is kind of paralleling what we're seeing in the KYNU and HAAO het mice for the sepsis study. The KYNU mice, knockout worms in red, are maybe a little sensitive to P. aeruginosa in these kind of intermediate ages, whereas the HAAO knockout worms maintain this ability to resist the pathogen later in life. And you can kind of see this day of exposure versus mean survival on this axis, where the HAAO worms have this later life ability to resist on the survival front. So it looks like we're getting some parallel to that here. And it looks like knocking out HAAO, we think it's through elevated 3-HAA, is benefiting in this case. We've recently made worms where, using CRISPR, the endogenous HAAO, we have an M. scarlet fluorescence protein attached. And so when we expose them to P. aeruginosa, HAAO does have a response. We were expecting maybe that HAAO was, something was going on with HAAO here. There is a slight upregulation in HAAO and maybe a change in localization. So there's something going on here. It's maybe not super dramatic, but we are getting some response to HAAO. And for some background, it's this first enzyme, IDO-TDO, that's known to be immune responsive. And these downstream elements, there's less known about them. And that's one reason we're focusing down here. But that looks like there may be some immune response throughout the pathway. But more strikingly is when you tag KYNU instead, which is, again, the molecule producing 3-HA, that's upregulating 3-HA, we see kind of this more dramatic upregulation in KYNU expression, and probably a change in localization as well. We're just starting to play with these models and kind of nail down what's going on here. And so we think that there's some upregulation of the relevant genes for producing and breaking down 3-HA. The other thing we can do, and we want to combine this with our HAAO and KYNU M. scarlet models, is we've tagged the bacteria with GFP, so in a different channel. And that allows us to do tracking. So if you look at kind of the worm, you can see early on when they're exposed to P814, you get a point infection, and this slowly kind of spreads throughout the worm's gut. And what we want to see is whether around these infections you see kind of a spike in this expression levels. The other thing we can do here is follow progression in these mutants, and this is still pretty preliminary, but one thing we see is that the HAAO worms later in life, they have a slower progression through these different levels. So complete infection, so the redder this is, the more severe the infection, and the HAAO worms are protected against that. And this is just quantified between like day 3 and day 11 of infection. The final thing that we're looking at is trying to figure out what 3-HA is doing potentially to increase this resistance. And one thing it might be doing is actually slowing the growth of the bacteria if you're increasing local 3-HA concentrations. And this is actually true, it seems, in both E. coli and P. aeruginosa, that the top, these are just growth curves. And as you push the dosing up, the growth of these bacteria go down. So at least one thing it looks like it's doing is that like a local 3-HA concentration may actually be directly inhibiting bacterial growth. We're also starting to look at the immune pathways in these contexts to see if we're getting activation of the various immune signaling pathways, which is the potential other side of this coin. So that's kind of where we're at. Again, the kind of next steps in this process is to look at the dosing for 3-HA and LPS. We want to do more work with these HAAO and KYNU knockout strains to see if we can see their resistance to LPS, and then eventually move on to bacteria present, and that's this CHiiLi ligation and puncture, where you puncture the gut and get bacteria in the interperitoneal space. You can also just directly inject P. aeruginosa as a second model. And then we have a metabolomics profile to look at what the pathway is doing in each of these cases. And then worms, we want to do kind of a parallel set of experiments. We want to look at how they respond to LPS to see if we can get a more direct comparison to the mouse models, and then continue using some of these fluorescence models to really dig into the details of where expression is changing and how it's responding to the aeruginosa infection. So I'll leave it there with that. These are the folks that are doing the work. Of course, Luis Espejo is a graduate student in the lab, and he's kind of leading the inflammatory work going on with the Kynurenine pathway, and the people involved are the other members of the lab who've been working on these projects as well. So I'll leave it there and say thank you and answer any questions.
Great. We do have a time for a couple quick questions.
So my question is for both you and Chris, actually, and maybe more for Chris than for you. Are any of these pathways lighting up in humans with sepsis that you have analyzed, Chris? Or George, have you gotten any samples from him?
No, not yet. I'll talk about it in a second. We have about 38 right now. The blood is still there. I didn't want to do, because of the batch effects, I wanted to reach at least a good, decent size. Because if you do it too early, then you lose a lot of your power, and then you start introducing batch effects. So I'll get with Josh, and then we'll see. Let me just give updates when I talk, but not yet. Yeah.
Sure.
Yeah, I'll second that. I think if we can run these as a batch, we'll get cleaner data. But yeah, we've got the metabolite profile. We can basically look at every metabolite in the Kynurenine pathway, all the way from tryptophan down to the immediate precursor to NAD. So we'll be able to look at those in the human samples once we've got them.
Yeah, Josh, just regarding the animal experiments, for my models, we use 1 milligram per kilogram. Mine is usually LPS, intratracheal LPS, and 1 mg per kg is a pretty heavy dose. So you're giving 5 or higher. That's a lot.
Yeah, are you looking at survival in your models, or are you?
No, it's lung injury, and these animals, they struggle. And now, granted, this is intratracheal, but that's just 1 mg per kg. So I'm assuming that if you wanted to do a survival model, you probably want to go even lower.
Yeah.
And then there's the issue of the volume as well. I mean, just imagine if you give a very high volume, that in itself is a problem.
Yeah, absolutely. And that's kind of what we're finding just based on these results, is that we get a very fast kind of short survival of these mice. And there's a couple of papers that we base this on where they looked at the like 2.5 all the way up to like 20 mg per kg of LPS in young mice to generate the kind of fairly rapid survival curves. But maybe I'll talk to you more offline, and we can that might help us nail down what the right dosing range for these models is.
Very good. Thank you. I think a nice transition. We're going to start with Dr. Chris Bim's presentation.
Can you see my screen?
Yep.
All right. Well, thank you so much, everybody, for attending. I will be giving an update on this project, looking at the clinical and immunologic predictors of frailty in the geriatric survivors of septic shock. And this project is thanks to the support that I've received from this wonderful collaboration. I'm thankful. As I will be updating you, you realize that COVID-19 has really been a major hindrance to our ability to get the sample size as quickly as we would have wanted to. So I'll talk about a little bit of background, how we came to this project, and really focusing on the effect of oxidative stress on aging and response to injury. And then I'll talk about the proposal that we made and give an update on enrollment. You know, we know that the US population is aging and that the elderly are at increased risk for septic shock and increased mortality, as well as increase in readmission rates. We have seen that significantly with the COVID-19 pandemic. It's really disproportionately affected the elderly. And we think that there's a combination of frailty as a major contributing factor. Specifically, a lot of preliminary data from translational research is pointing towards the effect of reactive oxygen species on the innate and adaptive immunity that is supposed to help fight this virus. And so the same could be said about the traditional causes of sepsis as we have understood over the years. COVID-19 has just made it even more glaring. And one thing that we found is that biological age varies in relation to chronological age. And there is a lot of heterogeneity, which complicates decision-making in critical care. So the fact that someone may be 80 years old doesn't mean they will not survive their critical illness. And, again, conversely, someone who might, be 50 years old might actually look much older or respond much worse to the kind of injury that we see in critical care. We think that there are genetic underpinnings to this immune response, and that is what this project was trying to explore. Some challenges with our sepsis. There's no perfect animal model. George just talked about sepsis. LPS injury is not really reflective of real life sepsis. If you think about it, we struggle with the amount of dose for different models. We struggle with the mode of administration of LPS. Is it IV, IP? Is it a cecal ligation? Or is it, in my case, when I focus on lung injury, I do intratracheal. And so translating that into other large sepsis included are usually very acute and have a very short time. So it's very challenging to investigate those. And then there's a lot of heterogeneity. And clinical risk factors poorly predict at-risk patients. So we know that oxidative stress is important in precipitating the worst outcomes that we see. There's always a balance between the antioxidant effects as well as the increase in oxidative damage from free radicals, which we think promotes inflammation. And the better way to see this is to look at, this example of an apple. That's just to show you how within a very short time, oxidative stress can break down the cells. And this is what we imagine to be happening in patients who succumb very rapidly to sepsis. Like I said, there's been a lot of work looking at trying to see if antioxidants, different types, could mitigate the effect of sepsis or septic shock. Unfortunately, most of these attempts have not been very useful. And the challenge is, how do you really study oxidative stress? There are so many outputs that could indicate oxidative stress. There are different organs respond differently, different injury models generate different types of oxidative stress, and the antioxidant responses tend to be different. So we wanted to look at more of, go upstream and look at the genetics and see what is happening at the level of the genes that are being upregulated or downregulated with respect to sepsis and reactive oxygen species. And this was an in silico work that we did, where we looked at microarray gene expression data from peripheral blood mononuclear cells in patients with sepsis. And we constructed the first reactive oxygen species gene expression based signature that was actually capable of predicting the odds of mortality. And we published this a few years back. And it was really an interesting work using translational research. We had two cohorts, based on data sets from the gene expression omnibus, where we looked at 26 survivors and 9 non-survivors in the discovery cohort. And we had in the validation cohort 78 survivors and 28 non-survivors. And you can see the details of their age distribution there. The survivors tend to be younger, obviously. And so when we looked at that, we were able to identify 450 genes that were upregulated and 1,027 that were downregulated. And we showed that based on this heat map, there is a clear difference in which genes are upregulated or downregulated based on the survival status of the patients. We proceeded to look at the enriched pathways based on these genes, and we saw that oxidative phosphorylation was the pathway that was most significantly associated with survival. And so we proceeded to look at the specific pathways that are involved. And again, generation of precursors of metabolites, energy translation, and oxidative phosphorylation. So we looked at the overlap between these genes and survivorship in sepsis. And when we drilled down, we found that there are specific 21 genes that overlap between, ROS-specific genes and sepsis survival in our sample. And again, these are these genes, again, looking at the different pathways that are involved. A lot of them include pathways that clearly implicate the antioxidant effects here, as well as just increase in oxidative stress. We proceeded to do a validation of this signature with the new data set. And basically this summarizes the fact that compared to survivors, non-survivors had a higher score. And when we combine that score, we're able to generate an area under the curve of almost 0.7, which is pretty decent. And then we did a random permutation with, just randomly generated gene sets. And when we compare that to our gene set, we showed that we had a very, very good chance of identifying survivors versus non-survivors with a significant p-value. So we felt very strongly about this gene set. So on this project, our proposal was to validate this 21 gene set predicting survival in elderly patients. So our goal was to recruit elderly patients in our ICU, where we would look at gene expression as well as biomarker analysis to see if we can do a combination of the gene expression as well as the protein biomarker cytokine profiling and generate a more robust prediction model. Again, this is important because, like I said earlier, a lot of the outputs that we use to measure oxidative stress, like MDA, are not very specific. And you have to have a clean animal model or cell model to maybe make a better prediction. But in the human samples, it becomes more challenging, especially if you're dealing with just blood samples in patients who have sepsis. So the hypothesis was that reactive oxygen species associated gene expression and this panel of inflammatory cytokines that are measured at the time of ICU and at the time of discharge would determine the frailty status among survivors of septic shock. And the plan was to see these patients as well as upper extremity frailty assessment with a non-invasive tool. We did that in collaboration with NEMA. And for specific M2, we would follow these patients up in clinic and then see how their frailty transitions occurred. We have a post-ICU clinic that where I would see these patients in clinic post-discharge and actually afforded the clinical benefit of being able to coordinate their care. So it was a good effort that was helpful for the patients. That's a timeline, just a little schema for our recruitment and follow-up. We had a lot of questionnaires. We did some baseline SOFA and Apache scores, and then we got the blood for cytokines and gene expression. And then, of course, we would follow mortality, ICU days, and other outcomes in terms of functional status post-discharge. So, we screened a lot of patients, and this speaks to the challenge of doing this kind of research. When I first presented this, it seemed like, just imagining the number of patients that I see in the ICU, that it would be easy. Well, it wasn't. We screened more than 1,000 patients, and this is really routinely looking at every chart. And for a very long time, only about 87 met enrollment criteria. And between trying to get the patient surrogates or tracking people down, we only got 38 of them that were consented. The average age was about 68 years, 20 males and 18 females. 15 of them died in hospital, all were sent to hospice. But we completed upper extremity frailty assessment on 21 of those patients, and I saw about 7 or 8 of them in the clinic. And we're hoping that, because like with most research that you do in the ICU, once you establish the processes that we had done, then it's a ramp-up phase. So I was hoping to, at the very least, reach the 60, 70 patient mark so that we can start having the kind of statistical power for this genomic or cytokine analysis. Otherwise, like I said, it will be a moot point. And then COVID-19 happened, which should have been an opportunity, but unfortunately, because of the anxieties and the concerns in being able to see these patients or send coordinators to recruit and collect blood samples, or even do the frailty assessments, we were pretty much shut down. And so for most of this year, we really haven't been operational, which is unfortunate, because I think that COVID-19 is a great opportunity to really definitively answer this question, given that it affects mostly those patients who are elderly. And again, this cohort is meant to be very well phenotyped. One of the very few critically ill patients that are well phenotyped with post-ICU follow-up. I'll tell you that I belong to the post-ICU interest group at the American Thoracic Society that also looks at elderly survivors of critical care. And even big centers like Vanderbilt, they don't have this kind of cohort. And so this is going to be a very unique opportunity. If we can just drive this sample size to about 100, 150, we will have a lot of very important data that can answer questions definitively. So, hopefully as things get clarified, I'll be able to get back in the game. We've already submitted to be able to see these patients. The challenge is that we can't bring them to clinic because there's a lot of concern for infection, but I'll keep trying to see if I can get some more of these patients. The other options would be to bypass some of the opportunities. Now, just like I said, these are things that are being looked in, and reactive oxygen species is thought to be a major driver of poor outcomes. The intersection with especially the innate immune system by leading to increase in neutrophil extravasation and formation of neutrophil extracellular traps that drive the cytokine storm, leading to multi-organ system failure and death. And there's also a feeling that these reactive oxygen species suppress T cell activity and worsen the outcome. Again, this is mostly speculative because a lot of the experiments have not been definitive, but it makes a lot of sense if you think about it. Again, in people who do not have the risk, there's a balance of pro-oxidant and antioxidant effects. In those that are at risk, an increase in pro-oxidant effects leads to a generation of more ROS, and then damaged red blood cells increase more cytokines and neutrophil extravasation, and then just drives this aberrant inflammatory response. So we have an opportunity to answer a lot of these questions, and I'm hoping that, you know, once I get to a decent sample size, we'll run the first batch of our analytes. The gene expression should be very easy, especially when you reach 50, because processing the PBMCs is challenging. So it's always good to have a decent sample size before you do that. Otherwise, the cost doesn't really factor in. For the cytokine profiling, I was very happy, thanks to Yanko, for allowing us to collaborate with the Garcia lab to buy a novel mesoscale discovery platform that can do so many cytokines at once. We can do upwards of 15 cytokines. That's much cheaper in a smaller sample of plasma. So we have that ready to go at any given time, and we shouldn't have a problem running a lot of analytes at once. And so that's where we are right now. And hopefully, as soon as possible, we will have more to share to the rest of the group.
Thank you. Thank you, Dr. Bimp. Now this presentation is open for questions. You know, the recruitment always been challenging, especially with older adults during the pandemic. I wonder if there's any way can we use a mobile unit to visit, to do a home visit?
Well, we could, but one of the challenges is that we need to do the follow-up upper extremity frailty assessment, because, you know, that is a major surrogate outcome. And so we couldn't do that remotely. We would have to have a waiver for the coordinator to visit the home. So that, you know, that we can explore that, but it will be a lot more challenging.
Yeah, it will be. But as you said, this is a great, actually, time window for study this. Yeah.
Yeah, I mean, I'm open to other suggestions.
Excellent. Any other questions? Good.
Sorry, I was muted. Just a very quick technical question regarding the mesoscale machine. Chris, is it realistically being available for use by, you know, some of in our community, or is it busy and really completely occupied right now?
So it's in our lab, so we can talk about the details offline. It just requires a lot of manpower, but I'm sure that if we can buy the assays for the analytes, we should be okay.
The reagents, right? Yeah.
Yes.
Well, that would be good. So we'll talk offline about that.
Right. My technician just did a lot of COVID samples, and sometimes they have to come over the weekend, but they can run 50 or 100 samples very quickly.
Great. Thank you.
Okay. Let's move to our next presenter. Dr. Mohab Abraham, you're on.
Thank you, Dr. Chen. Just sharing. All right, so what I wanted to talk with today and share with the group are my approaches to offer complementary management of post-operative pain. So some surgical facts about the volume of surgeries that's taking place worldwide. In 2012, there were over 300 million surgical cases. In the US alone, in 2010, there were close to 50 million surgical cases. And these were the latest statistics that I was able to find. The expectation is that the number of cases will increase with time. Over 80% of these patients report significant and severe post-operative pain. Currently, the most common post-operative pain management involves either NSAIDs or opioids of some sort, or combination of both. The problem with NSAIDs is that it can increase some types of post-operative bleeding. And with opioids, obviously, the side effects associated with it are quite known. In addition to that, especially in the elderly, there are additional side effects of opioids that can present as a significant inconvenience. For example, urinary retention, and that, of course, in addition to the respiratory depression. So the thought is decreasing opioid needs for postoperative pain would benefit both the patients and the hospitals, especially the elderly patients. In my previous research, I have shown that green light exposure was able to decrease some of the chronic pain conditions. Now, as you can imagine, postoperative pain is acute in nature. So the question is, will green light exposure work for postoperative pain? So this is just a summary of my previous data that was published about 2 weeks ago for fibromyalgia and migraine patients exposed to green light. So what we've noticed with fibromyalgia is that green light exposure decreased their pain intensity by about 40 to 50%. And for the migraine patients, it did two things. First, it decreased the intensity of their pain by about the headache pain by about 50%. And it also decreased the number of migraine days per month. So will that work for postoperative pain? So in this case, I used a rat model for postoperative pain. It's called the Brennan model, in which a vertical incision is made at the hind paw. And this model typically produces a hypersensitivity to both thermal and mechanical stimulus. And based on that, we can assess the success of our intervention. The model takes about 24 hours to develop, and by 48 hours, the model starts to reverse. So the rats will spontaneously recover after 48 hours. So in the first panel here, what you can see is almost a dose response, if you will. I'm talking about light, and I'm trying to apply some pharmacological concepts. So I will probably need some open-minded people here. So in that case, a dose response corresponds to the amount of light that the rats are being exposed to. So we expose the rats to either 0, 2, or 4 days to green light prior to the surgical incision. And what you see here, if you look at the bar graph, the white light, basically we measured the area under the curve, and that was our baseline because white light is used as a control. Now, the rats that were exposed to 2 days or 0 days prior to surgical incision, you can see that the area under the curve increased probably just a little bit. However, as the number of days increased of exposure to to green light increase, you can see a continuous development of analgesia. Now, if we look at the actual data itself, the black curve here is just the baseline. That's the normal progression of rat. And this is a power withdrawal latency. So this is response to a thermal stimulus in which a source of heat is focused on the rat paw. And we measure the time the rat moves away from that light source. The faster they move, the more we interpret this as more pain. A normal rat will respond to about 20 seconds. After surgery, they will respond to about 20 seconds. The response here being moving away from the heat source. And by 48 hours, they will start to reverse. Now, as you can see, the darker the green curve, that means they were exposed to green light more, or for more days prior to surgery. As we increased the number of days of pre-surgical exposure, we actually were able to reverse the effect of surgery. The same thing was seen, almost identical response for mechanical stimulus. So 4 days of exposure to green light almost reversed mechanical hypersensitivity by about 50%. Now, we wanted to see if green light will act in an additive or will potentiate the effect of morphine. And what we've seen, again, I will direct your attention to the bar graph here. And this was done for both thermal and mechanical stimulus. So here we measured the area under the curve, and we assumed that this corresponds to the amount of analgesia or reversal of the hypersensitivity of the surgical incision. And green light alone increased the area under the curve by about 50%. what exactly we saw in the prior slide. Now, 1 milligram of morphine, which corresponds to this white bar here, we assume that this is 100% of the area under the curve. When we added green light to it, that increased it by about 50%. So it potentiated the effect of morphine by 50%. And in fact, when we increased the morphine to 5 milligram alone, so now we're comparing 5 milligrams of morphine to 1 milligram of morphine combined with green light, we saw that 1 milligram of morphine combined with green light was actually more efficacious or more potent than 5 milligrams alone. So what we did here, we potentiated the effects of morphine and we lowered the dose needed, or the doses needed to exhibit its effects. The same thing was also seen in a mechanical stimulus, almost identical. Now, we wanted to explore a little bit as to how this is happening. So we looked at the CSF and we looked at two cytokines. The first is tumor necrosis factor alpha, and the second is IL-10. TNF alpha is inflammatory in nature. And what we have seen is that the longer the rats are pre-exposed to green light prior to the surgical incision, the less TNF alpha that has been expressed in the CSF, suggesting that we are managing this, or one of the possible mechanisms is through the cytokines. IL-10, on the other hand, did the exact opposite, which is great because IL-10 is anti-inflammatory. What we've seen in the CSF is that it has the concentration of IL-10 increased. So one of the sources for these cytokines are the microglia. So we looked at the IBA1 expression, which is a marker for microglia, and we've seen that with white light exposure, which is the control, we've seen significant increase in the staining of the IB1 marker. However, when we pre-expose the rats to green light, that when the IBA1 staining basically went back to baseline. So that means something is happening with the microglia. So we did a 3D modeling for the microglia. And what happened, if you look at the left lower panel, this is a microglia cell in the spinal cord for rats that were exposed to white light and underwent surgery. So this is the control. What you can see here is an enlarged cell body and small processes or ramifications. This is actually suggestive of activation of the microglia. However, when a different cohort of rats were pre-exposed to green light prior to surgery, you can see certain morphological changes indicated by a smaller cell body and longer ramifications. This is suggestive of microglia being less activated. So the message here is that exposure to green light prior to surgery decreases the activation of microglia in the spinal cord. We also wanted to see what happens with long-term potentiation. So we looked at two receptors, the NMDA receptor and the AMPA receptors, both of which are intimately connected. What we have seen is that there were really no change in the expression of one of the subunits of the NMDA receptor. However, there was significant decrease in one of the subunits in the AMPA receptor, again suggesting that the green light has long-term potentiation. Now, the next step is that, well, this is great, it works in rats, but will it actually work in humans? So that's the next step of the project. So stay tuned to see what's going to happen next. Obviously, a lot of people worked on this, and I would like to acknowledge all their support and all their help. In addition to the CHiiLi initiative, which was very instrumental, also from the Department of Anesthesia and Pharmacology and from the NIH. With that, I'll be happy to take any questions.
Thank you, Dr. Abraham. Great presentation. So I'm looking forward to see the results from the rats to human being. That's any questions for Dr. Abraham?
Mohab, awesome story as usual. Did you guys do? I mean, that may be difficult or whatever. Did you guys, or at least think of doing the experiments where you might sever the optical nerve to document that this is primarily coming through the visual pathway?
Very nice question. Actually, we just submitted an R01 grant looking specifically at your question. So this is the main question of that grant. There are two pathways for the light to get its effect, either through the cones and rods or through intrinsic photosensitive ganglion cells. These are not image forming, but they are light sensitive. We really don't know which pathway. Is it the optic nerve or the lateral geniculate pathway? So one of the question will be to lesion one of those pathways and see what the outcome is.
Cool.
Yeah. Because one of the criticisms we got when we published our fibromyalgia and migraine papers is that the visual spectrum for rats are significantly different for humans. So rats will see ultraviolet, but they will not see in the red spectrum. Where humans, so we don't know what rats see when we shine green light on them. So their perception is obviously something we cannot understand.
Excellent.
Great work, translational work, for us to better understand the mechanism as well as the application of green light. I know a lot of people, working in the pain field feels like we're really short of strategies to manage pain.
So one housekeeping point before I forget, for all of you, if you can send the updates on your funding that may have been related to the CHiiLi support to Amy and to Zhao and myself, that would be great. We will need this for reports for sure. And it's very important for Jennifer Barton and the TRIF part as well.
Okay, thank you. And we're going to move to the next presenter, Nima, Dr. Nima Tuzet.
Hello. Can you hear me good?
Yes.
Yeah, thank you. Sorry, I missed the first part. I was teaching in the class, but now I'm here finally. So yeah, I'm going to talk about the project. We actually finished this project, but we still in the stage of post-processing the data and getting more out of it. This is a continuation of a previous project we started a few years ago, and with the main goal of providing a device that can be used in the clinical setting for assessing frailty. And the main focus, of course, is to make it implicable for those who are in the emergency setting, for those who are going through the surgery. There's not enough time to do more comprehensive measures of frailty, and basically want to spend like 4 or 5 minutes with the patient to make sure we can collect this data and be able to use that for assessing the post-surgery complications that might happen to these patients. And for this, we are focusing on those who are going through major abdominal surgery process, and we're trying to identify those who are frail, and to understand how the post-operative complication after the treatment or during their hospital stay, and as well as discharge and readmission coming back to the hospital would be increased for those who are frail and identify as frail. So this was a very good opportunity for us to be able to test our device for the first time in the clinical setting where it's supposed to be worked. We tested this on patients outside the clinic, and we know that cross-sectionally it has a very good association with more common ways of measuring frailty, like free frailty, but we got the chance to test this on actual patient in the clinic. The main goal was to see how this measures of frailty pre and post treatment can predict the outcome. We're measuring the in-hospital outcome and post-discharge outcome. And of course, we also wanted to see, is it feasible to do this test on patients? And they were able to doing, because most of them would be bedbound and they're not able to walk. So we're, and with all the IV attached to them, we want to make sure we be able to do the test that we developed. So I'm going to talk about the design of this process, and then I explain how we do the measurement. So this was a longitudinal study, and we followed with the patients for a duration of 30 days, which happened through the phone calls and emails, however we can get in touch to the patients, which would be the most difficult part of this study. And then after the admission to the hospital, we go see the patient before they go through the treatment. Again, we had to capture this patient before they go to the surgery, which was also very difficult task to do. Most of the surgery done early in the morning, that basically there's not enough time to do this assessment. So we have maybe 5 to 10 minutes to spend time with the patient to get these measures before they go to the surgery. After that, we have more time. So we, in addition to the UEF, our measures, we were able to get other tests that are common to get from these patients after their surgery while they're staying in the hospital. And we get some outcomes during their stay, like how long they stay in the hospital and any surgery complication that can happen and the level of pain they tolerate during their stay. And through the phone calls after discharge, we measured, we assessed the readmission or any emergency visit that happened, or any complication that can happen to this related to their surgery or their the whole process of their stay in the hospital. And of course, we follow up with them to see if anyone dies through this whole process. Yeah, as I mentioned before, we would able to just quickly do our test, the UEF functional and cognitive test, and do some measure of their pain during the first visit. In the second visit, we have more time. We could do the MoCA test as a cognitive test, and the surgical risk score, pain, and the lifespan questionnaire. So I'm going to briefly explain the process of the measurement of the UEF. I talked about this before in other presentation, but if you have more questions, I would be happy to answer at the end. There are two tests that we're doing. One is to measure the physical frailty, and the other one is to measure the cognitive frailty and the level of their cognition. For the first test, we asked them to do a quick 20-second elbow flexion while they're equipped with the wearable sensors attached to their forearm and the upper arm. And based on this test, we have developed this app that we can quickly measure several measures that is related to physical frailty, such as the slowness of their movement, how flexible they are, how much they get tired, what are the muscle forces that is activated during this task of elbow flexion, and how much they get tired during this 20-second task. So those changes and those measures are very subtle, but using this technology of wearable technology, we can really measure them and give them some objective values. And that's what we're doing here. In addition to that, we have developed a cognitive aspect to it, because several different studies, they show that there's a high association between physical frailty and the cognitive status of the person. And we wanted to, for the first time, come up with one single test that can provide both the physical frailty and the cognition status of the person. And what we are doing here is to use the dual test surrogate for this, and asking the person to do the motor function and simultaneously doing a cognitive task. and we look at the trajectory of the motion, and based on that, develop some outcome that is related to the cognition status. This has been a very well-established approach for gait assessment, gait dual tasking, but it's never been done for the upper extremity. And we're the first people developing this. And we really see that it works the same way as the gait test does. And it's easier to perform, and there's no need to have a place to walk, or especially for these patients that are in the bed, we can quickly do it in the bed. And again, we developed this app that quickly, right away, can post-process the data and eventually give a score from 0 to 1. 1 is for the physical function, and then the other one is for the cognitive function. So I tried to list some of these parameters that we extract from this test here, and there are a bunch of them, and we keep adding more parameters to this to make it more, just adding those that really realistically predict the condition of the patient. For example, if you think about the physical frailty, we more focus on dyspnea, sarcopenia, how their muscle performance is. When we are looking for the cognition, we look at the motion irregularity, and then how they execute their motor function while they're doing the dual task. So we put all this together and give this to a score. We got outcomes. We've completed the data collection for 100 participants. Unfortunately, there were some missing data points, both for the first visit and second visit. Sometimes we're not able to catch the participants before they go through the surgery, and sometimes they're lost to the follow-up. But we eventually managed to get 100 participants. And I tried to list different reasons for the admission here. The mean age was 61, and about 4 days they stay in the hospital on average. 2 people unfortunately died, one during the hospital stay and one after discharge. And you can see a huge number of readmission, almost 30% readmission for this participant. Although we missed some post-discharge outcome, you see a huge number of readmission that happened through this 100 participants that we recruited. So very quickly, I want to summarize the result we got. Here in this figure, I'm showing our scores that we have for the UEF and how it's different from those who had any in-hospital complication and those who didn't. So the way I'm presenting this result is to separate them for in-hospital outcomes and then post-discharge outcome. You can see there's a huge differences here between the score we give to these groups. Green having no complication and red having complication. And there's still significant difference with age, but what we get from our score is more predictive compared to the simple age of the person. So we try to put this in a model to see if we can predict the outcome. And we able to get to this sensitivity and specificity in terms of predicting the outcome in hospital outcome when we use our score. Especially when we merge these two scores, we come up with a very nice surrogate to measure these complications. And then we moved on and tried to develop the same model for the post-discharge outcome. Again, the same results. It was really interesting to see age was not significantly different for those who showed adverse outcome after discharge, but our scores showed the difference. So really, age by itself might not be the best way to say who might go through this kind of complication after the treatment. Again, we used our model and tried to predict the outcomes, and we got pretty nice area on the curve of close to 0.9 when we use these measures that we have for physical and cognitive function. And you can see, if we just use age, it's pretty much like a random number. It's 0.5. It's very close to predicting nothing when we're just focusing on age. But doing these two tests, we were able to improve this accuracy by almost 40%. So this has been done, and we already finished the post-processing, and we also finished the first paper, which we submitted to we're going to submit today, actually, to the BMC Geriatrics. And but this is not over. We believe there's so many other things we can do with this data. We just do a simple model to show that our device is basically working for predicting the outcomes. But there's other ways to do the post-processing of the data. We are working with other teams to do machine learning, to just looking at the raw sensor data, the time series, to be able to develop more robust ways to calculate to predict the outcomes. And this is an ongoing project. Hopefully we are finishing this in near future. We'll be able to submit another paper based on this data. And this was a very good platform. I'm thankful for the CHiiLi program to provide this opportunity. We were able to, for the first time, to show that this device is working. And now it's time to go for a bigger project to increase our sample size in the first place and improve our modeling approaches and go with a longer duration of post-operative follow-up with the patient, because 1 month might not be enough. And we're trying to get that done and submit another grant, hopefully soon in near future, in R1 based on the study. And the main goal is to promote this device, and we know that it's working, and we just have to, do the advertisement and make sure that we can not just use it in the banner, but also use it in other hospitals to make sure, because now it's purchased by Best Buy and they're producing it. So it would be pretty much available for everyone to use. And I want to thank everyone who helped out for this process. All the students that spent so much time in the hospital to be able to get these patients, and also the CHiiLi program for the funding source. Thank you so much. If you have any question, I would be happy to.
Great, Nima, very, very, very impressive data. I think set up the stage to move forward with many different type of studies. So I want to ask first question, and then people can jump in. So Nima, how do you think of this? First of all, what kind of equipment, the sensor you use? Second, whether you think that this is a very outcome specific, or can we expand it to other patient population?
Yeah, so for the first question, we're just using the already developed devices. So we started with a set of sensors that are basically the gyroscope and accelerometer. We want to say that it works. So now we know it works. We develop our own app. The device is just as specifically being produced for the upper extremity. So it's going to be something specific for this test that has been out there to purchase. And again, this is the gyroscope and accelerometer, but is designed to just focus for the upper extremity test. And to answer your second question, yes, that's exactly what we're trying to do. We're not trying to focus on one sample of participants. This was just like an example of that, showing that this device is working. But if you think about the frailty, just using it as a more generic term, that you can apply this device for every single patient that is admitted to the hospital. That would be the ultimate goal. Not looking at what causes this kind of frailty, but really measuring, because we're not focusing on what is the underlying disease that caused this frailty. We want to say that this person is frail, even if it's because diabetes, because of the abdominal pain, or any kind of disorder that can lead to that. But make it more generic. That's the goal for the next R1 that we want to write about this.
Congratulations.
Thank you.
Well done. Any other questions for Nima? We were planning to do this seminar in July, but just because of the pandemic, we thought it was better to respond to the pandemic first, and then we'll come back to talk about our research. I was just really impressed with everybody's work. Keep up with your research during this very hard time for both your research and the participants. So congratulations to all. So Yanko, do you have any final remarks to the group?
Not really. I'm just very thankful to everybody for sharing wonderful data. It was fun. As I said, we just need information on any papers and grants obtained from the beginning of funding, just so that we can keep track and justify the investment. And other than that, if there's anything that you can think of where we could help you further, try to overcome some of the COVID-19 obstacles or shutdown obstacles or whatever we can, let us know.
I have a quick question. So for papers that come out of this funding, if you can provide a grant number or any number they can put in acknowledgement, that would be great.
Yes. Yeah, we'll send you a standardized formulation that everybody should use.
Perfect.
Any other questions from presenters or audience? If not, I wish everybody a wonderful Thanksgiving and be safe if you get together with your family or friends. So the pandemic is still going, so I hope we'll see each other in person soon. But if not, we'll definitely bring back everybody to present in the future virtually. So it's really great to see all you on the screen. And thank you again for a wonderful session.
Thank you.
Bye now.
Thanks, Zhao. Thanks, Amy.
Yes, with somebody working behind the scene, making this seamless for our presentation. So Amy, take a bow.
Oh, thank you. No worries. I'm glad to do it.
Wonderful job. And all the
Thank
help.
you, everyone. No,
I know.